Aš LMA kačiulis.
Gerai, ir keliu. Čia į viršų.
Ir viskas. Esam pasiruošę. Saugu. Saugu. Gerai, ačiū labai.
Tai va, įkėlė. Numeris. Tik tai...
Jo bus?
Jo. Kačiulis. O kiti kur nežinau?
Kačiulis, nu tai čia jie patys žino. Oi.
Baigėt žaist šitą dienotvarkę.
Labą dieną, prašome sėsti. Pradedame Lietuvos mokslo akademijos visuotinį susirinkimą. Ir šis susirinkimas yra įspūdingas, kadangi bus pasirašyta sutartis su Kazachstano mokslo akademija. Ir bus inauguruoti 3 užsienio nariai ir, žinoma, jaunųjų mokslininkų stipendijos ir kiti svarbus dalykai. Taip, kad pradedame visuotinį susirinkimą ir pirmas klausimas, tradicinis, darbotvarkę matote ekrane. Ar pritarėte tokiai darbotvarkei? Tai bendro sutarimo pritarta. Ačiū. Na ir pradedant posėdį, šis posėdis bus vykdomas 2 kalbom, lietuvių ir anglų, ir kazachų. So, Your Excellencies Ambassadors, dear guests, this meeting will be in 2 languages, in Lithuanian and in English, and in Kazakh language as well. So we have a chance to learn Kazakh language, and I apologize, prominent guests, that part of this meeting will be in Lithuanian. So we are starting this meeting, and first of all, it's a sad news. Our one foreign member passed away, so I ask to have a minute of silence. Minutę tylos pagerbti buvusi mūsų narį. Ačiū. Na ir pradedam prie malonių dalykų. Norime pasveikinti vieną akademiką, kuriam garbus amžius, gimtadienis. Tai gerbiamas akademikė, Lietuvos mokslo akademijos prezidiumas ir technikos mokslų skyrius sveikina Jūs gražaus jubiliejaus proga. Nuoširdžiai dėkoja Jums už aktyvią veiklą Lietuvos mokslo akademijoje ir technikos skyriui. Žinomas mokslininkas, produktyvus, linkime jam sveikatos, sėkmės, naujų kūrybinių idėjų. Sveikiname Vytautą Ostaševičių. Dabar jaunųjų akademijų. Na ir kitas malonus dalykas, tai yra jaunųjų mokslininkų stipendijų pažymėjimų įteikimas konkurso nugalėtojam. Ir pradedame nuo socialinių humanitarinių mokslų sričių.
Taip.
Prašom.
Lietuvos mokslo akademijos prezidiumas 2023 m. birželio 20 dienos nutarimu numeris 25 Lietuvos mokslo akademijos jaunųjų mokslininkų stipendijas paskyrė šiems jauniesiems mokslininkams. Humanitarinių ir socialinių mokslų srityse daktarė Kristinai Razmienė, Vilniaus Gediminos technikos universitetas, vykdomų tyrimų tema žiedinės ekonomikos ir žinių perdavimo sąveika siekiant įvertinti ES valstybių narių darnaus vystymosi potencialą. Daktarui Tomui Rimkui, Klaipėdos universitetas, vykdomų tyrimų tema priešistorės technologijos, kaulų ir augo dirbinių gamybos technologijos ir chronologija akmens amžiuje Lietuvos teritorijoje. Nedalyvaus, negalėjo atvykti. Daktarui Justui Stončiui, Klaipėdos universitetas, vykdomų tyrimų tema Lietuvos tarptautinis įvaizdis 1988-2022 metais, lietuvių ir žydų santykių veiksnys. Gamtos medicinos ir sveikatos, žemės ūkio ir technologijų mokslų srityse. Daktarui Tadui Dambrauskui, Kauno technologijos universitetas, vykdomų tyrimų tema funkcinių kalcio hidroksilatų su įsiterpusiais geležies ir kadmio jonais sintezė ir savybės. Daktarė Greta Jarackytė, Nacionalinis vėžio institutas, tyrimų tema mezenchiminių kamieninių ląstelių ir fotonų energiją aukštinančių nanodalelių teranostinių galimybių tyrimas. Daktarė Vitailėlė, Lietuvos sveikatos mokslų universitetas, vykdomų tyrimų tema galvijų priešpinių biologinio ir cheminio profilio modeliavimas, taikant tvarius sprendimus efektyvesniai galvijininkystei. Daktarui Pauliui Mackoniui, Fizinių ir technologijų mokslų centras, vykdomų tyrimų tema pažangių didelio intensyvumo vidutinio infraraudonųjų spindulių ruožo lazerio architektūrų kūrimas. Daktarui Arnoldui Pautieniui, Lietuvos sveikatos mokslų universitetas, vykdomų tyrimų tema erkinio encefalito viruso paplitimo Lietuvoje analizė laike ir erdvėje bei prognostinio modelio kūrimas. Daktarui Simonui Ramanavičiui, Fizinių technologijų mokslų centras, vykdomų tyrimų tema 2D MHNU nanostruktūrų pritaikymas biomolekulių aptikimui panaudojant paviršiaus sustiprintą Ramano spektroskopiją. Daktarė Mildai Stankevičiūtei, Gamtos tyrimų centras, vykdomų tyrimų tema automobilių padangų didelių dalelių ir monospecifinių parazitinių infekcijų ekotoksikologinio potencialo lašišinėms žuvims tyrimai. Negali atvykti. Daktarė Gintarė Šidlauskaitė, Lietuvos agrarinių ir miškų mokslų centras, vykdomų tyrimų tema daugiamečių pupinių ir miglinių žolių variacijos tyrimai žolynuose, siekiant pagerinti agrarinės aplinkosaugos ir derliaus stabilumo rezultatus. Daktarui Mantui Šimėnui, Vilniaus universitetas, vykdomų tyrimų tema fazinių virsmų naujose maišytose hibridiniuose perovskituose spektroskopiniai tyrimai. Daktarui Justui Šlaitui, Vilniaus Gedimino technikos universitetas, vykdomų tyrimų tema linkiamųjų gelžbetoninių sijų su nemetalinė armatūra įstrižojo pjūvio elgsenos analizė. Daktarė Gretai Verkalaitė, Lietuvos sveikatos mokslų universitetas. Vykdomų tyrimų tema – laisvai cirkuliuojančio DNR mutacijų profilio analizė sąveikų, atrofinių gastritų ir skrandžio vėžių sergančių pacientų kraujyje. Daktarui Mantui Žiauniui vykdomų tyrimų tema – aplinkos poveikis baltymų amiloidinių fibrilių struktūros įvairovė ir jų sąveikai su amilodofilinėmis molekulėmis. Nedalyvaus. Ačiū.
Pakviesčiau visus laureatus padaryti bendrą nuotrauką. Ateikite čia su pažymėjimais.
Pranešimą tarptautinio bendradarbiavimo plėtra Lietuvos mokslo akademijoje skaitys prezidentas Jūras Banys.
Gerbiami kolegos, gerbiami svečiai, jeigu leisite, kad būtų įdomu, aš šį pranešimą perskaitysiu ir angliškai, kad mūsų svečiai suprastų. Ar nebus prieštaraujančių? Okay, so dear ladies and gentlemen, I will give a very brief and short overview how the Lithuanian Academy of Sciences, what is doing in sense of international cooperation with other institutions in the world. So, speaking about present situation, of course we must first return a little bit back in history, and in 1990s the first agreements have been signed between Baltic academies and Swedish Academy, and this was the first international agreement. Then Lithuanian Academy of Sciences became a global player. And the interesting thing that foreign members, especially today we have inauguration of 3 foreign members, it was introduced only in 1990 as a foreign members by the statute of the Academy at that time. And Academy consists of 114 today. The limit, you know very well, is 120. Today we have 114 full members, 59 emeritus members, and 61 foreign members. And this number will change soon after this meeting. It will be additional 3 members, so it will be 64. Academy not only participating in various, I would say, associations, European level, but also organizing a lot of international conferences, events, and it's worth to mention when Lithuania was chairing European Union, part of the events have been in the Lithuanian Academy of Sciences. Then Lithuania was president of the Council of European Union. International associations is a huge role, starting with all European academies, LMA, European Academy Science Advisory Council, ESAC, and Science Advice for Policy by European Academies, which is very important voice in European Commission, where we can do evidence-based advice to European bodies, European Commission, and this is very important. We have to mention also that was European structures. We take active part also in inter-academy partnership. Which is global and covers whole world, from United States to Japan, South America, and so on. Also, another global structure which we are actively taking part is International Science Council. And also we have active activities, or a lot of activities in Federation of European Academies of Medicine and Union of European Academies for Science applied to agriculture, food, and nature. Also, Academy have a lot of bilateral agreements. We have to mention, in 1990, it was signed agreement with Royal Society, so British Academy of Sciences, or English Academy of Sciences, better to say, because in Britain they have Scottish Academy, Welsh Academy, and this is Royal Society's English Academy of Sciences. And one of the last, it was 3 Baltic states, Lithuania, Latvia, Estonia, and in Germany, Hamburg Academy of Sciences. We also have agreement, so dear Professor Franke, you can participate also in that scheme. So if you look at the map, it's to list all, but you can see European map, all the agreements with 26 foreign academies. Also important to mention, Lindau Nobel laureate meetings. We are choosing the candidates to go to this meeting. And 3 academies, Armenia, Azerbaijan, and Israel, is not on European map, but we also have agreements with these academies. Due to these agreements, a lot of people are visiting Lithuania, so incoming and outgoing visits, and you can see the statistics, how many days per year in different years, so from 2018 statistics till 2022. This year has not been yet finished, but we had guests from Hungary, Poland, Latvia, and so on. So you can see that a lot of visitors to Lithuania, to different research institutions, universities, institutes, libraries, and so on. Another important issue, Academy of Sciences by President Rudzikas initiated cooperation with CERN. And now this cooperation has been moved to consortium of universities and institutes. So Academy still active part, and I can mention that 25th, 26th of September, so next week, will be CERN Commission examining Lithuania progress in work with CERN in nuclear particle physics. And this evaluation can lead to the full membership. Now Lithuania is associated member. Full membership, of course, includes a bit more fees, a little bit higher, but we will see how it goes and are we suitable to fulfill full membership conditions. Also, foreign members from time to time, please, dear colleagues, who will be inaugurated today, do not forget Lithuanian Academy, and from time to time, guests are visiting Academy and giving lectures and being in touch with Lithuanian Academy of Sciences. So, foreign members, we can see that Germany is far from ahead of all other countries. That's the statistics of foreign members. And you can see that it's very broad spectrum, starting from Germany, United States, United Kingdom, Poland, Sweden, and so on. And of course, we have Canada, Iceland, Japan, Netherlands, Norway, China, and Spain. And also we are very proud that our 2 foreign members are Nobel Prize winners. Gerard Mourou, he got his Nobel Prize for work in laser physics, and Robert James Schiller from United States, who received Nobel Prize in Economy. And they gave lectures in the Academy. Another approach, instead of personal meetings and personal lectures, due to pandemic, everyone got used to Teams, Zoom, WebEx, and all of the different programs. So a section of biosciences, they started remote meetings with foreign members, discussing various issues, and that was very useful. So other sections, I think they will do the same. We have to mention close collaboration with Baltic academies, with Latvia and Estonia. Each 2 years we have a meeting which changes the topic, and I am very glad that the last one, which was in Vilnius, it was devoted to genes, genetics, and it was very successful. Next one in Tallinn was devoted to mathematics for society, big data, and all that stuff you know very well. It's becoming more and more important. And the last one, this spring, was in Riga, and in Riga was devoted to energy. Due to the war, you know very well, electricity, renewable energy, and such issues, they are very important. And of course, after this meeting, usually there are declarations. So totally statistics that from representatives from our academy took in so many meetings in LMA, International Science Council, ESAC, IAP, and so on. You see the statistics. I will not go into details. And what's important that our members of Academy are giving a significant contribution to statements, studies, reviews of important problems, starting from nutrition, food, ecology, green energy, and so on. And these are publications from these umbrella, what we call umbrella associations, which are published in a recent year. Another, you remember, I hope, that we have Young Academy, and Young Academy is also very active in international field, and they are World Young Academy, and also taking part in different events of young academies, especially in North Europe, Scandinavia, and we hope that we will host Young Academies Baltic Sea region Young Academies meeting in Vilnius soon. Another outreach is that Lithuanian Academy of Sciences is publishing scientific journals, so you can see that these journals are reaching different libraries in different countries, and 221 issue of periodicals are sent to different countries. Of course, Poland is the leading one, 42 copies, and so on, and different countries which can read what our researchers have published. Another international cooperation we have with international scholarly societies. It was a philology society, and here is the example of physics, European Physics Society. They have a tradition to put a commemorative place of the lab, which made a significant contribution to the science. And as example, I can say that in in Bern is Einstein plate, in Copenhagen Niels Bohr plate, and in Latvia, Estonia, and Finland, they do not have this, and we succeeded because it's a strict evaluation. Theodor von Grotius was evaluated due to the charge transfer mechanism that he invented. In his hometown, it's a statue and plate of European Physical Society. That this is marked place. Another, we work internationally with UNESCO and Women's in Science, and we are very glad that Eva has reached not only Baltic State Award, but also, I am afraid Eva is here, so she will correct me, it was a world, European. But nevertheless, never it happened to Latvians and Estonians, only in Lithuania we have 2 European level laureates. So, another, we have close relations with Ukraine Academy of Sciences, and due to the Russian aggression, war, thank you very much all members of Academy. You donated the money and 2 generators last winter. When it was tough with electricity and heating, it was sent to Ukraine, to Academy of Sciences, because I spoke with the president of Ukraine Academy of Sciences, asked what we need, and the answer was we need generators. Not computers or whatever, and after that, this year we sent also 2 servers to Academy of Sciences. So thank you very much for supporting Ukraine. And this my brief talk, and I will be happy to answer your questions if you have some. Thank you very much.
Are there any questions, comments? No? Thank you, President, for information.
Thank you. Taigi, tęsime toliau. And now, very important moment, we will sign agreement between Kazakh Academy of Sciences and Lithuanian Academy of Sciences. And due to that, I ask dear Excellency President Zakaria, please, the floor is yours. Please tell us how Kazakh Academy of Sciences look like.
Құрметті Латвия ұлттық академияның президенті профессор Банис мырза, құрметті ұлттық Латвия академияның академиктері, құрметті қонақтар.
Dear President of the Lithuanian Academy of Science, Professor Banis, dear Academy members and guests.
Сіздердің назарларыңызға Қазақстан Республикасының ғылымының негізгі басымдықтары туралы айқындап айтқымыз келіп тұр.
Today, on behalf of the National Academy of Science, under the President of the Republic of Kazakhstan, it is a great pleasure and honor for us to be here and present to you our country. And our presentation is devoted to the main priorities of the science of the Republic of Kazakhstan.
Қазақстан Республикасында ұлттық академия 1 маусым 1946 жылы құрылған. Оны құрған әйгілі біздің академигіміз Қаныш Сәтбаев. Осы жылы 27 наурызда, яғни 2023 жылғы 27 наурызда Қазақстан Республикасының президенті жанындағы ұлттық ғылым академиясы ерекше мәртебесі берілді.
The Academy of Science of Kazakhstan was founded in 1946 year, and by academic Qanish Satpayev, and after that by our President Kassym-Jomart Tokayev, Academia reorganized and the increase of the status and prestige of Academia in the 2022 ceremony.
Бүгінгі күні Қазақстан Республикасында ғылым басқарудың жаңа моделі жүзеге асыруда. Яғни, бүгінгі күні Қазақтың ғылым академиясы, ғылым бағыттары негізгі 10 ғылымның басым бағыттары бойынша жүзеге асырылады. Ғылымда 414 ғылыми зерттеу институттар және университеттер жұмыстарды жасайды. Және де бүгінгі күні көп пайыз біздің ғылым зерттеулер жаңа модельге ауысқан.
Today, the initiative to introduce a new science management model is being implemented in Kazakhstan. And as I mentioned above, the National Council for Science and Technology under the President of the Republic of Kazakhstan established. 10 priority science directions have been identified. 414 enterprises carrying out scientific research and experimental design are performing their activities.
Негізгі шаралардың біреуі бұл бүгінгі күні ғылым және технологиялық саясат бойынша жаңа заңнаманы қарастыру, және де Қазақстан Республикасы жанындағы үлкен ғылыми кеңестердің және сараптама орталықтарын жұмысын жеделтіру. Оның ішінде бүгінгі күні Insights рейтинг бойынша Қазақстан 65-ші орынды қамтып отырады. Ғылымның ішінде әйелдер пайызы, яғни ғылыми әйелдердің пайызы 52%.
And currently, the development of the new law of scientific and technical policy, and we are in the pre-foresight stage of conducting foresight and analytical research of the development of science and technology. And we implemented the program popularization of science among students and young scientists. And also at the same time, we are pleased that Kazakhstan ranks 65th in the Insight system, and importantly, 52 of the total number of scientists are women.
Қазақстан Республикасында ғылымдар, ғылым қызметкерлері, ғылыммен шұғылданатын 22 мыңнан астам. Оның ішінде зерттеуші мамандар 18 мыңнан астам. Солардың ішінде 46% жас ғалымдар, яғни 40 жасқа дейінгі ғалымдар. Осының ішінде ғаламдық қор аясында ғылыми зерттеулер 2500-ден астам асырылып отыр. Қазақстан Республикасында өте үлкен ғылымдарға бағдарламалар өтіп отыр. Оның ішінде негізгісі жас ғалым гранттары, бұл халықаралық грант, 1000 орындық гранттар, және болашақ бағдарлама бойынша 500 халықаралық тағылымдар бойынша гранттар жүзеге асыруда. Оның ішінде үздік зерттеуші 50 грант беріледі.
And of course, due to new politics, we supported scientists and intellectual potential of our country, and based on the figures of 2022, more than 22,000 scientific workers, 18,000 researchers, and 46% of young scientists under 40 years of age are currently working in Kazakhstan, and 2,529 scientific research projects are being supported with direct state funding. And every year, young scientists has an international grants and more than 2,000 researchers. And 500 scientists are given the opportunities to take 3 to 1 years international internship. In addition, 50 best researchers awards are given to the best scientists.
Қазақстан ғылымының соңғы 5 жылындағы негізгі жетістіктермен таныстырып кетеміз. Оның ішінде Қазақстанда COVID-19 қарсы вакциналар 5 платформада әзірленген. Оның ішінде дүниежүзілік денсаулық ұйым үміткер вакцина тізіміне енгізілген инактивациялық Казвак вакцинасы Бүгінгі күні заводтың ішінде 5 миллионнан астам доза шығарылып, бұл вакцинамен Қазақстан мен Қырғызстанның азаматтары егілген, және де 2 векторлық вакцина әзірленген, 2 суперлік вакцина және тірі аттуинеттерген вакцина шығарылған. Яғни 5 платформада 5 түрлі вакцина және де барлығы ВОЗ-дың тіркеуіне кірген.
In last 5 years, scientists in Kazakhstan have achieved some results. And firstly, dear guests, Kazakhstan vaccine of COVID-19, and you can see here our president of Academy of Science, Kunsulu Zakaria. She's the one who created vaccine Kazvak. This vaccine, one of the vaccine who WHO is take to the list of vaccine against COVID-19.
Екіншісі өнімділігі жоғары биоақпараттық енгізу жүйесі Q-симфониясы, транскриптомалық, экзомдық және биомедициналық деректерді талдауға арналған Q-симфония жоғары өнімді биоинформациялық есептеу жүйесі.
The next achievement, that creation of Q-Symphony high-performance bioinformative computer system, and this system designed for the analysis of high-dimensional genomic, transcriptomic, exome, and biomedical data.
Сіздер білетін шығарсыздар, Қазақстанда өте жоғары дамыған ядролық физика. Соның ішінде токамак реакторы, жабық магнитті тұзақ болып табылған. Біреген тағамақ ядролық кешені жоғары температуралық плазманы жасауға және оны ұстауға арналған. Бұл термоядролық реакция жүргізуінде мүмкіндік бүгінгі күні берілген.
The third significant scientific achievement is a tokamak reactor. This unique tokamak nuclear complex closed magnetic trap. It is designed to create and contain high temperature plasma. This made it possible to conduct a thermonuclear reaction.
Бүгінгі күні энергия үнемдейтін инновациялық шамдар шығарылған ғалымдармен шамдар энергиясы айрықта үнемдеу арқылы жарықты 2 есе артуын қамтамасыз етеді. Плазма мен нанобөлшектер синтезін кванттық қасиеттерін зерттеу осы патенттелген пайдалануға дайын өнімді жасауға әкелді.
The fourth achievement is innovative energy-saving lamps. The lamps provide double the brightness with significant energy savings. Research into the quantum properties of plasma and nanoparticle fusion led to the development of patented, ready-to-use product.
Электромобильдердің соңғы жылдары көлемі артқан, соған қарай электркөліктерін зарядтау станцияларды біздің қазақ ғалымдары шығарылған. Электрлік көліктер зарядтауға үнемдейтін технологиясы шетелдік аналогтармен салыстырғанда зарядтау құны айрықлармен төмендетіледі.
Next achievement, our achievement are charging stations for electric vehicles. The cost-effective charging technology of electric vehicles significantly reduce the cost of charging compared to foreign counterparts.
Отандық Қазақстан қорытпасы, яғни этнокартография бойынша Ватикан, Италия, Австрия мемлекеттерде Қазақстан қорытпасы және материал металлургия болатын тоқсандыру деректерді және модификациялау үшін қолданылады. Бұл жерде кішкене шатастырдым. Айта беріңіз.
And another best achievement is the creation a book atlas about the Kazakhstan state of European and American maps of the 19 centers. A unique book atlas dedicated to depicting the Kazakh state on European and American maps of the 19 countries resulted from 15 years of research.
Супераккумуляторлар, бұл зерттеулер патенттелген, өте тез бірнеше минутта зарядталатын суперконденсаторлар, сонымен қатар супераккумуляторларды шексіз директоры зарядтау және разряд циклдері бар. Кәдімгі конденсатор үшін бұл ерекше тек 1000 және суперконденсатор үшін 10.000-нан 5000-ға дейін циклге дейін, яғни олар 10-500 есе көп болып тұр.
Next, our achievement super batteries. These super batteries can be charged very quickly within minutes, and super batteries have an almost infinity charge-discharge cycle for the ordinary capacitors.
Және де сіздерге әйгілі Қазақстанда бүкіл Менделеев таблицасына кіретін элементтер өте көп. Қорғасын және алтын өндіру үшін жаңа инновациялық технологиялар ашылған. Әлемге бастапқы қорытынды әрбір 10 тоннасы біздің технология арқылы өндіріледі.
And you know that in Kazakhstan we have a lot of elements from the table of Mendeleev, and we use this to our technology.
Және де Қазақстанда өте көп мөлшерде мұнай геологиялар бар. Мұнай өңдеу процестерінің катализаторы шығарылған радий және иридий қосылыстары мен фосфор бар ионды сұйықтардан негізделген мұнай өндірудің инновациялық катализаторларды шығарылды.
Catalyst of the oil refining process, we also use that important innovation of oil processing innovator discovered by the Kazakh scientist.
Беттік акустикалық толқындар шексіз күшейту әдісі бойынша Қазақстан және ақш зерттеу физика графенді пайдалана отырып, аз энергия жұмсай отырып жоғары жиілікті акустика сигналдарын күшейту туралы жұмыстар өткізіліп, бұның ішінде өте жақсы негіздер және патенттер графиндік пайдалануға мүмкіндік берілді.
And researchers from Kazakhstan and the United States have discovered in physics that they can amplify high-frequency acoustic signals using graphene, using less energy. This discovery allows the use of graphene, the strongest, thinnest, and lightest conductive material in micro and nanoelectronics.
Және де қазақ мемлекеті үшін ерекше картография бойынша кітап шығарылды, кітап Атлас. Бұл 16-19 ғасырлар европалық және америкалық карталарда қазақ мемлекетін бейнелеуге арналған бірегей кітап Атлас шығарылды. 15 жылдық зерттеуінің нәтижесінде бұнда Европа және ақш-та табылған 300-ге жуық орталық ғасырлық карталар табылып шығарылды.
And as we mentioned about that unique book, Atlas about the Kazakh state, devoted to 16th, 18th centuries, created, and it is because that due to our big research.
Бүгінгі күні Қазақстан Республикасының ұлттық ғылым академиясы жастармен үлкен жұмыстарды атқарып отырады. Сол жұмыстарға жастарды тартуда. Сараптамалық топтары елімізде 1000-нан астам жас ғалымдар тартылған.
And of course, our academy is working with the young scientists, and more than 1,000 young scientists from the country were involved in expert groups.
Және де 120-дан астам жас ғалымдар сала алар жобаларды жүзеге асыру жұмыстары жүргізілуде. Ғылымның басым бағыттары бойынша 10 жұмыс тобы құрылды және ғылым, инновация, іскерлік, ынтымақтастық жүзеге асырылуда.
And implementation of interdisciplinary project with more than 120 young scientists in the country, and 10 working groups were created on priority areas of science. Science, innovation, business cooperation is being implemented.
Құрметті Литва ұлттық академияның президенті Бонис мырза, сізге шақыруыңыз үшін үлкен рахметімізді айтып, біздің жұмыстарымыз әрі қарай ғылым саласын жетілдіріп, ортақ дәрежеге жеткізуге апарат деп сенімдіміз. Көп рахмет.
Dear President of Lithuanian Academy Nauk, Professor Bonis, dear guests, dear members, thank you very much for attention, and of course we are ready to take and making joint projects, and thank you for your invitation. Thank you.
Рахмет.
Thank you very much. Now we proceed to the most important part.
very much. So now we have chance to go to Kazakhstan and do research there. And you know Kazakhstan is a very rich country. Have whole periodical table of elements. So geology, geography, history, please use the chance. And also physics, medicine, all fields are interesting.
Taigi, artėjam prie kitų svarbių įvykių. Šiandien toks susirinkimas, vien tik svarbus įvykiai. Tai užsienio narių inauguracija. Ir pradedame nuo Lietuvos mokslo akademijos užsienio nario, profesorio Andrė Franke, biologija, Vokietijos Federacinė Respublika, inauguracijos.
Diploma Membri Hospitis Academia Scientiarum Lituania. Academia Scientiarum Lituania in convento universali dia quarta aprilis anno Dominis bismillesimo vicesimo tertio prongentibus meritis ergas scientiam atque pro auxilio Lituaniae benigno tributo referendisum Dominum Andre Franke Respublica Federale Germania professorem doctrinae biologice membrum hospitem Academia Scientiarum Lituania elegit. Professor Juras Banys, Presidents.
Sind sehr froh, dass wir haben noch ein Mitglied aus Deutschland und das zu beweisen, Sie kriegen Diplom, das hat Vizepräsident gerade gelesen, das ist in Lateinisch, ich hoffe, dass als Doktor Sie können Lateinisch perfekt und auch sehr wichtig, Sie kriegen ein Stück von Gold. Bitte schön. Thank you very much. So, you can congratulate. So now the floor is yours.
Yes, thank you very much. It's a great honor for me to accept this membership. I felt some pressure when you mentioned that you have 2 Nobel Prize winners among your list, just a little bit of pressure. So I'm a geneticist by training, so I will talk about genetics, and I've been in this for over 20 years. And it's not only about freedom you mentioned, writing manuscripts, writing grant applications, great ideas. It's also about great collaborations and friendship, okay? And I just wanted to illustrate in the next minutes where this can take you, to interesting paths. At least in my career, it has been really interesting which roads you sometimes take. So up here you see our research center in Kiel. It looks like an unknown flight object in gold that landed. And we have here in the basement, we have a great biobank where we store biosamples for mainly genetics research. And up here we have some high throughput laboratories. So I'm from Kiel. I think most Lithuanians know where it is because there is a ferry commuting between Klaipėda and Kiel. And these are the instruments that we use a lot, okay? So these are sequencers, DNA sequencers, with which you can decode DNA, okay? And we were fortunate in Kiel that we started on very early, okay? So it started in 2007 that we bought the first instruments here. Some of you may know the Roche 454 instruments still. The first individual genomes were really made with this machine, also from Watson, for example, and other prominent persons. And we invested a lot over the years, okay? It's really a key technology. It's like a Swiss Army knife these days because you can do a lot of it, yeah? DNA decoding is only one part of it. You can also look at gene expression patterns and so on. And that's why we are also participating in several genome projects, okay? And I'm coming back to my first slide. It's interesting to see where the road is taking you. So we hired a postdoc who worked in New Zealand before. And so he brought in a lot of collaborators from New Zealand and also some interesting samples for which we do the reference genomes. And I just wanted to show some highlights here. For example, I was collaborating with Andreas Keller from Saarbrücken on microRNAs, okay? And he introduced me to the working group on the Eisman. You know maybe that Eisman is the oldest mummy that has been found, that is in great shape still for research. We call it Ötzi in German. It was found by two mountaineers in 1991. And there was a big fight whether it's an Austrian mummy or Italian mummy. I think it was first transported to Austria and the pathologists looked at it and they didn't do any proper things to avoid contamination there. And in the end it ended up in Italy, and you may look at this in a museum in Bolzano, okay? It's a really good museum. I highly advise to visit it. It's all about Ötzi, the Eisman, and the mummy. And we actually helped to resequence the genome and to analyze it. And thanks to the data, we know that the Eisman had brown eyes, it had blood group O, he was lactose intolerant, he probably also suffered from Lyme borreliosis, and so on. So there are a lot of things you can read out of an individual genome. Yeah, here's again the mummy. We later on also sequenced the stomach content. So then we understood what the Eisman was eating. We also tried to reconstruct the microbiome, but that didn't work out, okay? So the microbiome looked more like the microbial environment of the glacier. And in the end, the biggest success here was when the data was reanalyzed by Ben Krause-Kuora in our institute, that they reconstructed perhaps the oldest Helicobacter pylori genome for the Eisman. Fun fact, I hope this is not the only thing you will remember from my talk. Brad Pitt has an Eisman tattoo. I didn't know this. These are the things you learn in the museum in Bolzano. The other interesting project that we are working on and that we have just submitted is the reference genome of the reindeer. Okay, why the reindeer genome? Well, a great colleague and friend meanwhile, Tom Hemming-Carlsen, he's a hepatologist in Oslo. He's also a big reindeer hunter, okay? So he goes out and hunts reindeer, and because he's so much interested in these animals and because of other political issues, we decided to sequence the genome, okay? So he's sampling here, and to make something bigger out of it, we sequenced different reindeer populations, and a particularly interesting one is the Svalbard reindeer, okay? So this is the smallest reindeer species around from Svalbard, and it's very small, okay? So it's about 20–30% smaller than the normal reindeer. And this is Santa Claus reindeer, okay? Because it's the North Pole there, Svalbard, and so I didn't know that before, that these are actually quite small reindeer. And we looked at the genome of especially the Svalbard reindeer, compared it to other reindeer, And we looked at genes that were under selection, that carried a lot of interesting mutations. And to make a long story short, we found a lot of genes that have been implicated also in humans in ciliopathies and other diseases that give rise to body malformations and height reduction. Especially here in the centromere machinery, we see that a lot of these genes and proteins are acting, and these genes have been implicated, as I said, in human diseases like the Seckel syndrome, kind of dwarfism. So it makes sense, and it may explain why these reindeer are smaller. And the other thing that we found are also that there are genes involved in motility and function of the respiratory cilia. And you need this in the Arctic, yeah? You need to breathe differently, and it's also known, I think, from other animals living there. So it all makes sense, and it all started with a crazy idea with a good friend. Now, these are just some side projects, okay? And we mainly worked on complex diseases and identifying causes of these complex diseases. It's much more than genetics, meanwhile, I will come to that. And we also worked on COVID-19, yeah? But the really focus here of our group is genetics of inflammatory bowel diseases, okay? So chronic gut diseases. And so far, we know 235 susceptibility regions in the human genome that are relevant for IBD. And we didn't stop since then. There are large studies ongoing. We found 174 new. They are not published yet, so we know over 409 loci in the human genome. And that completes really the genetic risk map here. And most of these genes, they point towards a disturbed interaction of the host immune system and the bacteria, so our own microbiome. So something is out of balance there, and so that's why we decided early on also to analyze the microbiome, okay? And just by looking at the numbers, you can see that we processed a lot of samples for many, many different projects in the microbiome field. There are a lot of publications resulting from these analyses, and we're currently extending this also to other populations in the world. And we just hired also 2 new young persons from the MIT who are setting up this bioreactor in Kiel. And let's see where that road will lead us to. Now, in the last part, I just wanted to focus on another story that has just been accepted. As said, we are interested in IBD, and here the HLA or MHC proteins are very important because they present environmental peptides, for example, to the immune system. Here in yellow are the unknown peptides, the unknown antigens that are disease-causing. And you have the T-cell receptor. And in that story, we particularly looked at CD4 positive T cells. So a young professor that we also recruited to the institute, Petra Bacher, she established this so-called antigen reactive T cell assay. Okay, so you can take blood from patients and you can test if their T cells react towards specific antigens or lysates or substances. And to make this long story also short is we tested different candidate bacteria and we didn't see a differential response really if you compare healthy people and Crohn's disease patients. But if we look at lysates of fungi, yeah, especially rarer Candida species or yeast, Saccharomyces cerevisiae, we see that the patient's T cells, they give a really, really severe and different response. And it turns out that these T cells, they are cross-reactive and they are also toxic, okay, so they can damage the epithelium and they are cross-reactive. And we even know that different cheese and everything that contains yeast can activate these cells. So we don't know yet if this is really causing the disease. At least it could be another mechanism why these patients have a chronic disease and get a regular relapse, okay? And we also see already in relatives of patients, so first-degree relatives that are at higher risk, that these cytotoxic cells are already there at increased levels. And this has just been accepted and been a great success. So this also puts now fungi into focus in our research, apart the microbiome, and that's something that we want to follow up in the next months and probably also years, because it really needs integrated approaches to identify disease causes and to identify disease-causing antigens. For us, this is like finding the Holy Grail, and you need some crazy people that share your enthusiasm to find this Holy Grail, to really find the disease-causing agent in this severe disease. And for this, we also now have a EU program that is jointly coordinated with people here in Kaunas, and where we look at early disease, pre-disease cohorts, and try to find actual causes of disease. So this has started this year, and I'm very much looking forward to this project. In general, it was really a joyride with you here in Lithuania and the group in Kaunas. You can see here there are a lot of joint publications, many of them in Nature Genetics. We also have Nature here, and so on. We even made it to a cover here of Nature Genetics. This was driven by Yogita when she was still in Kiel. She's now professor in Kaunas, so very successful work. There are a lot more manuscripts. This is, I think, just an extract from our working group here. We also have plans for other infrastructure projects, how to collaborate better. For example, to create an excellence center. So there are different grant opportunities that we are pursuing here because we share the enthusiasm on inflammatory diseases, and there are many similarities here and strengths and weaknesses on both sides that we can exchange. So, I'm coming to the end. Thank you very much. It's been really a joyride, and I thank you here as friends. And I really liked the road that we took so far. Thank you very much.
Thank you. Questions, please. Like in the conference, now it's time for questions. I do not see any, so thank you very much. Now we proceed further. So, please.
Diploma membri hospitis Academiae Scientiarum Lituania. In conventu universale dia 4 aprilis, anno Domini bisilesimo decesimo tertia, pro ingentibus meritis erga scientiam, arche pro auxilio Lituania benigno tributo, referendissimum Dominum Abdul Mounzen, Regnum Belgicum, Professorium Scientiarum Agriculturae, Membrum Hospitem, Academiae Scientiarum Lituania elegit. Professor Juras Banys, Presidents.
new member of the Sales Academy. And now, but later we will make each other. Okay. Now the floor is yours.
Good afternoon, everyone. It is my privilege and honor to become a member of the Lithuanian Academy of Science and represent my country, Belgium. I don't know if I am the first member or I am the second member from Belgium coming and become a member of the Academy. I am from Ghent University, Faculty of Bioscience Engineering, and I am leading a laboratory on precision scoring. What I will try to talk about today is to give an overview of some history of my career, then present some main research domains, what I have done, and also focus mostly on findings in precision agriculture, and try to summarize where we are today and what the future will bring to me and to my team. So I have a long journey, really. I flew everywhere. I started from Syria, where I got my master and bachelor in agriculture engineering, and then I moved to Hungary, where I obtained a PhD degree in agriculture engineering, and then went back to become an assistant professor and an associate professor in Aleppo University in Syria. Then I was invited to become a postdoctoral researcher at Catholic University of Leuven in Belgium, where I spent about 7 years. And then I moved to the UK, where I was also invited to become an associate professor in Cranfield University for 9 years. And back to Belgium again, where I became a full professor in Ghent University, where I was awarded 3.5 million euros to create a center of precision agriculture in 2017. And lately I was also become a part of the team of the Vitatus Magnus University in Kaunas, and Professor Egidius arranged me to have a part-time contract as a senior researcher. So I'm very glad to be around here as well. I feel when I come to Lithuania every time very relaxed, and that's what I said yesterday to one professor in Kaunas. So the main research domains I have done in my career is about numerical modeling of agriculture soil-machine interaction. I did some considerable work in the use of spectral technology in different domains, in food, in agriculture, in natural resources, and lately in precision agriculture. And that's why I will also give some flavor of some examples of the use of multi-sensor data fusion in precision agriculture. So as you see here, this is the first when I started working as a PhD student and a postdoc, where we used numerical modeling like the finite element method and discrete element method to model the interaction between different tillage tools and soil, and that gave us an insight of the parameters affecting the design of these tools, and we were able to optimize the design of these tools to reduce the energy consumption and improve and maximize the quality of soil preparation. And we did that for different tools, and including tillage and also mechanical weeding. When we talk about sensor technologies, and this is our main domain, we do not use on-the-shelf products. We buy the basic spectroscopy, for instance, but then we engineer it we add to it different sensor technologies in order to make it usable for the domain we are interested in, and that is agricultural engineering, where I am standing today. As you see here, this is a penetrometer. Actually, it is cheap. You can buy it for 2,000 euros, but it is not as clever and smart as it should be. So we made it clever and smart. We added to it several sensors. Some of them are the optical sensor. You see inside the penetrometer we have optical fibers, and that is linked to visible and near-infrared spectroscopy. We can measure several parameters in the soil, including organic matter and compaction and moisture content. And we also attach to these sensors some frequency domain reflectometry. So as you see, it is a story of multi-sensor data fusion. Then you have a smart sensor that can measure those parameters that the normal commercially available sensor cannot. Then this is another multi-sensor data fusion kit where it is internationally patented, and here we put the spectroscopy behind this cutting tool in the soil. You put visible and near-infrared spectroscopy on a framework behind a tractor. You drive about 4 to 5 kilometers per hour, and you collect data every second about several soil properties like organic carbon, moisture content, calcium, magnesium, potassium, and phosphorus. So instead of having 1 point per field, which is the traditional way of soil sampling, we are able today to do 500 samples with high accuracy. And here where we come to an international patent on this system, which were our main sensor to use in our research. Then because we are interested in the agriculture system, we should not focus only on soil, but we should also have data on other parameters. And there where we have also again used and crafted commercially available sensors to acquire data on crop parameters. So we have soil parameters, we have crop parameters, and these all, together with topography and weather conditions, should be put into the modeling system in order to quantify the yield limiting factors. And when this is possible, then it's like you go to the doctor and you ask him, I have a pain in my shoulder. Then they start testing your blood pressure, your urine, and they do a lot of laboratory analysis to know what is the problem causing this pain. This is the same for the crop, but the crop does not speak. So it is even more complex. What you have to do is to collect data on several parameters affecting the crop behavior, and then you know what is causing the problem. Is it shortage of phosphorus in the soil? Is it shortage of water, or it is a disease affect the plant? And that is where we try to collect the multi-sensor data fusion approach. And then lately we put this sensor technology on robotics, and as you see, it is able to measure now while moving in the field without human intervention. And not only about soil sensing, we use this robotic platform in order also to collect data on crop and also to do control. As you see behind this robotic platform, we are able to do seeding or spraying of pesticide. For the first time ever, our group was able to manage one field without human intervention, from sensing to tillage, seeding, spraying, and even yield estimation. So this is where we see the future. It is sensing, modeling, and control, and everything should be automated in order to maximize yield, minimize the input, and reduce the environmental footprint, where we focus today. So let's give you an example of precision agriculture. Precision agriculture, really, it is to manage the spatial variability in the field. You see in this example, this is the actual situation in the 99% of fields in agriculture. You have always variability, but this variability means that you have here different levels of phosphorus and nutrients and moisture. That's why you need to manage this spatial variability differently. And while this field is like imaginary field, you cannot find it in actual world. So most of these fields are like this. And here we have to use this sensing, modeling, and control technologies in order to manage this spatial variability. And we use the 5 principles of precision agriculture. As said earlier, we aim to maximize yield and reduce the use of input in agriculture. So I give you a very nice example, and that is practical example. I love to tell it every time. We were in the UK in the field, and we measured with our sensor the phosphorus content. And we found here high concentration of phosphorus. And we asked the farmer what happens. He said, oh, I come here with my manure machine, and I put manure because here it is nearby the road. And that makes huge spatial differences, which is not good for the crop. So we said, please don't do that anymore. So we went in 2012 again, and we scanned the field, and we found indeed he was not putting manure anymore in this part, but we found an increase in phosphorus in the other corner. So we asked him what happened there. He did not admit that he put anything. He said, I don't know. Luckily, we were able to talk to his engineer, who said, yes, this time he went up to that corner and added manure in order to check if your sensor worked properly or not. So, which means farmers are very clever. We should not foolish them and never sell them technology that does not work. So when we used precision agriculture technology, we applied phosphorus where it should be according to the UK guidelines for fertilization, and we come to more or less more homogeneous application of phosphorus. Then we took this one step forward. We have everything is fully automated. We have a machine, a tractor. We have the sensor in front of a maize planter from a German company, Amazone. We can measure phosphorus in the soil, and in real time, we can control the amount of phosphorus fertilizer applied in real time. As you see here, the sensor in the soil transfer the data into a spectrophotometer, and then everything is being fully automated in order to control the amount of phosphorus applied. And here we have increased yield of maize by 336 kilograms and reduced the amount of phosphorus applied. We do the same multi-sensor data fusion approach for nitrogen fertilization, where we can compare, as you see here, we can collect several layers of soil data together with crop data and yield, and then we can fuse those into a map that is called fertility management zone map. And this can be divided into different strips where we can compare between precision agriculture application against the traditional way the farmer does. And we found in the first example that we can increase the profit to the farmers by 88 euros per hectare. But most interesting to say that if one sensor being used to measure 7,000 hectares over 4 years of time, this one sensor can save our society of about 156,000 euros, because this sensor can reduce the amount of nitrogen being dumped into the environment, and because of that will save money from the policymaker to clean our water resources and our soils. And who is paying that? This is our taxpayer, so we are paying this money. But one sensor, as you see, can make a lot of impact. We are talking about a lot of money. Not those only can go to the pocket of farmer, which I said 90 euros per hectare per year, but also we can save the society of paying a huge amount of money over 4 years of time. We can do that the same thing for spraying of pesticide. As you see in here, just the same example. We have a sensor, hyperspectral camera can be put on a spray boom. We have the soil sensors, and the data from the soil and the crop and from satellite can be fused in order to derive management zone maps for site-specific or precision spraying of pesticide. What we were able to do is do the same multi-sensor data fusion for manure management. Manure in Belgium, Holland, North Italy, Spain, in Catalonia, and also in Germany, is produced with high amount. And this manure, farmers do their best to inject as much as possible manure in their soil. And that causes environmental problem, because the soil is saturated with phosphorus and nitrogen. And that creates really environmental issues like nitrate leaching and phosphorus runoff. Luckily in Lithuania, I heard this is not a problem. In fact, in Lithuania, you will lack of manure to be used on the farm. So here where the precision agriculture technology can come and say, apply manure where the soil lacking those nutrients. In Belgium, I think it will be very difficult to find, but maybe in Lithuania will be very more likely to do this, and then apply this manure according to the crop need. And here you can see we have increased yield by 2.3%, we increased profit by 2%, and reduced the amount of NMP by 4 and 7% respectively. We took the technology one step forward, and we put the soil sensor in front of a tractor, and to the back of it there is a maize planter. So we can measure soil fertility by the sensor and give an order to the planter to apply the right amount of seed. So we precision seeding. There is a lot of waste of seeds that you should not apply in that rate. And you apply the amount of seed according to soil fertility measured directly. And by this, you could increase the profitability by 91 euros per hectare, which is money. And then, as I said before, we can do that with robotics platform, and we did that for potato seeding. So in the previous examples, I give it for maize seeding. This is for potato seeding. And you cannot imagine, when my PhD showed me those numbers, I did not believe it. He came up to 450 euros profit to the farmer. And that's something we should recognize. Summing up all these numbers, when we use this precision technology, we are really making profit to the farmer and reduce the environmental footprint. So increase profitability, reduce the environmental footprint, and at the same time have social impact. Not only using this for seeding or pesticide application, but can you combine all those together? As you can see for the example shown in here, where also profitability is very clear. So all in all, there was no time for me to really show all the research we have done in the last 25 years or more, but I would like to summarize what we have done so far. So my group has published 24 journal papers in Web of Science record with more than 8,000 citations, 46 H index. We have presented 150 conference presentations. We have 3 international patents. We have 10 keynote speeches. It is for me to do in international conferences, 1 full book and 30 book chapters. I have supervised 15 master students, 20 PhD students to completion, and 10 postdocs so far. I am associate editor of 6 international journals. I have secured more than 40 million euros grants and coordinated more than 10 projects. 5 of them are European projects, and also I have created a Center of Precision Agriculture in the UK. I was one of the 3 PI doing so, and now I'm working on a Center of Precision Agriculture in Belgium. So what are the future? The future is toward really pushing to adopt these solutions by farmers. If farmers does not use them, it will be blue sky research, and that is something I don't like. I really like that, to bring those into practice. I continue to do sustainable research with impact on society, and you see here we have hosted a demonstration to the Flemish Minister of Agriculture, Mrs. Krivits, in 2021, and she has seen how this high technology can be implemented in practice, but now the question is taking this technology into the society, and the farmer can be used. Hopefully not only in Belgium, but can be extended to other countries. I should really push to collaboration with my friends in Kaunas, as well as within my group. We are about 25 people from 13 different nationalities all over the world. And lastly, I will be very happy and supportive to the Lithuanian Academy of Sciences in the future. Thank you very much for your listening.
Thank you very much. And now, traditionally, questions. Are there some? No questions. Thank you very much for very interesting talk, because you mentioned global climate changes in agriculture is becoming more and more important, how we will survive, what we will eat. And thank you for an interesting lecture. Thank you very much. And now we proceed to the last foreign member. I do not know, I should speak English or Lithuanian. Tai yra medžiagotyros specialistas Saulius Kačiulis.
Diploma membri hospitis Academia Scientiarum Lituania. Academia Scientiarum Lituania in convento universali dia quarta aprilis anno dominis bismillesimo vicesimo tertio progetibus meris erga scientiam acte pro auxilio Lituania benigne tributo referendi sum dominum Saulius Kačiulis, Respublica Italiana, professorium scientiam materiarum, membrum hospitem Academia Scientiarum Lituania elegit. Professor Juras Banys, Presidents.
Lietuviškai.
So to be a guest, now is the next talk will be in Lithuanian language.
Man sakė, kad yra vertimas tiems, kurie nesupranta. Gerbiamas prezidente ir gerbiami kolegos, pirmiausiai norėčiau jums visiems padėkoti už man pareikštą pasitikėjimą išrenkant mane akademijos nariu. Toliau pabandysiu labai glaustai pristatyti savo veiklą medžiagotyroje, tiksliau paviršiaus analizėje ir indėlį į medžiagotyrą. Prieš pradedant kelionę po kietųjų kūnų paviršius, reikėtų trumpai apžvelgti, nuo ko prasidėjo mano mokslinė veikla. Ji prasidėjo nuo Puslaidininkių fizikos instituto Vilniuje, kur, kaip ir dauguma instituto darbuotojų, aš tyrinėjau karštųjų elektronų reiškinius. Karštieji elektronai tai yra didelės energijos elektronai, to pasiekiama stipriam elektriniam lauke. Ir pagrindinis mano tyrimas, kuris buvo pabaigtas su daktarinė disertacija, tai buvo karštųjų elektronų pernešimo reiškiniai 3-5 grupės puslaidininkiniuose. Tai buvo daugiausiai galvanomagnetiniai reiškiniai, magnetovarža, Holo efektas, ir sėkmingai pavyko pristatyti disertaciją. Tuoj pat po disertacijos, gal po metų, mano disertacijos vadovas, tuometinis Mokslų akademijos prezidentas Juras Požėla, man pasiūlė stažuotis paviršiaus analizės srityje, nes jis buvo labai įžvalgus kaip prezidentas ir norėjo sukurti Puslaidininkių institute paviršiaus analizės laboratoriją, pakankamai modernę, ir tiesiog atidaryti naują tyrimų kryptį. Jis jau tada numatė, kad karštųjų elektronų tematika kada nors išsisems. Taigi, nuo 85 prasidėjo mano kelionė po kietųjų kūnų paviršius. Aš ją pradėjau vėlgi rekomenduotas prezidento Požėlos, pradėjau prie Maskvos, Černogolovkoje, kur yra kietųjų kūnų fizikos institutas. Tai buvo turbūt stipriausia paviršiaus analizės laboratorija tuometinėje Sovietų Sąjungoje ir jai vadovavo, tarp kitko, irgi lietuvis, buvęs Sibiro tremtinys Vytautas Gražulis. Aš pas jį stažavausi pusę metų, toliau tęsiau savo pažintį su paviršiais Italijoje, Romos universitete, Tamperės universitete Suomijoje ir keliuose geriausiuose paviršiaus analizės centruose Jungtinėje karalystėje. Po to 88 metais sukūriau paviršiaus analizės laboratoriją Puslaidininkių fizikos institute. Aišku, padedant prezidentui Požėlai, nes tam reikėjo daug lėšų, daug rekomendacijų ir panašiai. Tačiau laboratoriją pavyko sukurti, jinai veikia, turėjom pirmus rezultatus iš šitos tematikos. Vėliau, 91 metais, išvykau į Italiją, kur gan ilgai su įvairiais kontraktais buvau vizituojantis profesorius, su Italijos grantais, su NATO grantais ir taip toliau. O nuo 97 metų jau dirbau paviršiaus analizės laboratorijoje kaip pastovus darbuotojas Italijoje. Taigi toks mano kelias į paviršius ir nuo tų 88, sakysim, metų mano pagrindinė sritis buvo paviršiaus analizės metodai. Dabar, kodėl tiek daug dėmesio tiems paviršiams ir kuo jie įdomus? Tai čia labai trumpa suvestinė, kad visos medžiagos sąveikauja su aplinka per savo paviršių, tas yra savaime aišku. Visos cheminės reakcijos, kurios vyksta ant kietuose kūnuose, irgi vyksta ant paviršiaus. Visi tokie mechaniniai reiškiniai, sakysim, mechaniniai kaip korozija, trintis, kuris sukelia susidėvėjimą, visą tai nulėmė kietojo kūno paviršius vėlgi. Ir jau nereikia nei kalbėti, kad bidimensinės naujos medžiagos, naujos kartos medžiagos yra tik tai paviršius. Dėl to paviršiaus analizė yra labai svarbus dalykas medžiagotyroje. Dabar reikėtų labai trumpai pasakyti, kaip suprantamas tas paviršius. Jūs matote čia skalę, matavimų ar tyrinėjimų, kuri prasideda nuo toli gražu ne nuo paviršiaus, o paviršius tai yra kažkur tarp 1 ir 10 nanometrų, tai yra pirmieji monoatominiai sluoksniai. Ir reikia iš karto įvertinti, kad jeigu skaičiuosi matomus, kiek jų yra visame tūryje ir kiek jų yra paviršiuje, tai santykis yra 10-8. Tai reiškia, kad įprastiniai matavimo metodai, kurie leidžia ištirti tūrinės charakteristikas, visiškai netinka paviršiui, nes kurti prietaisus, kurių jautrumas būtų 10-8 ar dar mažiau, yra be galo sunku, sudėtinga ir galbūt net ir neįmanoma. Reiškia, metodai turi būti visiškai kitokie. Čia tik tai šiaip savo dėl įdomumo prisiminti reikėtų, kad garsus teoretikas Wolfgangas Pauli, kuris Nobelio premijos laureatas, jis kūrė kietųjų kūnų teoriją ir jis apibrėžė paviršių kaip velnio išradimą. Tai labai suprantama, nes visa kietųjų kūnų teorija yra pagrįsta tūriu, visos formulės skaičiavimai ir taip toliau. O ties paviršiumi viskas baigėsi, viskas nutrūksta. Tai ir teoretikų požiūrių, ir turiu pasakyti, ir eksperimentatorių požiūrių, tai yra tikrai velnio išradimas ir reikia velniškų metodų tyrinėti paviršių. Čia yra pagrindiniai metodai paviršiaus analizės. Tai yra fotoelektroninė spektroskopija, OŽ spektroskopija ir antrinių jonų masės spektroskopija. Tų metodų yra daug daugiau, čia jų nevardinsime ir apie juos nekalbėsime, bet šitie 3 yra pagrindiniai ir jūs matote lentelėje, kuo jie skiriasi, kaip sužadinamas signalas. Pirmi 2 tai yra elektroninis spindulys, antrinių jonų masės spektroskopija, aišku, yra joninis spindulys. Toliau visi parametrai šitų metodų ir raudonai yra išryškinti tie parametrai, kurie nusako metodo pranašumą. Iš viso to labai gerai matome, kad pats geriausias, daugiausiai žadantis metodas yra fotoelektroninė spektroskopija, kuri leidžia daryti ir kiekybinę analizę, ne tik tai kokybinę, ir kuri suteikia informaciją apie medžiagos ne tik tai elementinę sudėtį, bet apie cheminius ryšius tyriamoje medžiagoje, paviršiuje. Bet viskas priklauso nuo medžiagos, kartais praverčia ir SIMS metodas, nes jo jautrumas yra labai didelis. Kai kuriais atvejais, kaip matysime vėliau, praverčia ir OŽ elektroninė spektroskopija. Čia kitos lentelės, kur jūs matot išvardinimą visų sričių, kur galima taikyti šiuos metodus. Ir su pažymėjimais, kuris metodas tai sričiai labiausiai tinka. Ir vėlgi, jeigu žiūrėsime į fotoelektroninę spektroskopiją, jūs matote, kad tinka beveik visoms medžiagoms. Tuo tarpu OŽ spektroskopija ir SIMS, jie tinka kai kurioms, toli gražu ne visoms medžiagoms. Čia yra nuotraukos iš mūsų laboratorijos Italijoje, kurioje šiuo metu turime 2 spektrometrus, vieną labai seną, bet jame yra ir fotoelektroninė ir OŽ spektroskopija, ir spektrometras veikia iki dabar, modernizuotas, pakeičiant elektronikos sistemą, kompiuterį ir panašiai. Kitas yra naujas, kuriame atliekama fotoelektroninė spektroskopija, ultravioletinė spektroskopija ir elektronų energijos sklaidos spektroskopija. Anksčiau turėjome laboratorijoje ir SIMS spektrometrą, bet jis paseno, sugedo ir mes jį prieš kokius 5 metus likvidavome. Kai mūsų turimų spektrometrų neužtenka, nes reikia, taip sakant, matuoti cheminius žemėlapius medžiagos, cheminę mikroskopiją atlikti, turimi metodai yra riboti. Jų skiriamoji geba nėra pakankama kartais. Tada keliaujame į sinchrotroną Italijoje Trieste, kur yra fotoelektroninė mikroskopija, leidžianti daryti cheminius vaizdus, ar cheminė mikroskopija su labai didelė skiriamąją geba iki 50 nanometrų. Jeigu to reikia, keliaujame į sinchrotroną. Čia yra išvardintos pagrindinės tematikos, kuriuose dirbau per šituos metus, per 35 maždaug metus paviršiaus analizėje. Ir skliausteliuose jūs matot publikacijas kiekvienoj tematikoje. Pirmoji tematika labai susijusi su mano darbu Puslaidininkių institute Vilniuje, nes toliau tai buvo puslaidininkiniai sluoksniai ir struktūros. Ir, kaip matot, nemažai darbų dar buvo iš to, bet ta tematika pamažu išsisėmė, nes tapo industriniu reikalu, o nebe moksliniu šitie tyrinėjimai. Toliau labai plati tematika, tai yra dujų sensoriai ir biosensoriai, kurią pradėjome iš karto po puslaidininkinių medžiagų ir kuri tęsiasi iki dabar. Labai plati tematika yra metalurgija. Tai reiškia metalų lydiniai, dangos, įvairios kompozicinės medžiagos ir kaip matot irgi iš publikacijų, tai labai daug darbo joje atlikome. Toliau, biocompatible, tai net nesakau, nežinau kaip pasakyti lietuviškai, biosuderinamos medžiagos turbūt. Tai reiškia medžiagos, kurios gali būti naudojamos medicinai, įvairios dangos, įvairios nano dalelės, kompozicinės medžiagos nano dalelėmis. Ir čia irgi indėlis buvo pakankamai didelis. Kiek mažiau tai buvo super laidininkai, super gardelės, bet irgi labai įdomis rytis. Retųjų žemių junginiai ir metalinės nano dalelės katalizė. Čia irgi nedidelis indėlis, bet irgi įdomi tematika. Ir paskutiniu metu daugiausiai dėmesio skyrėme, tai mažos darbo funkcijos medžiagoms, ploniems sluoksniams, kurie naudojami hibridinėse, termojoninėse, fotoelektrinėse celėse. Vėliau aš net pristatysiu vieną pavyzdį iš šitos tematikos. Ir plati tematika, tai yra bidimensinės medžiagos, tai reiškia grafenas, anglies nanovamsdeliai, fulerenai ir taip toliau ir taip toliau. Ir reiktų pastebėti, kad žemos darbo funkcijos medžiagos labai persikloja su bidimensinėmis medžiagomis, nes dauguma medžiagų su žema darbo funkcija yra bidimensinės iš tikrųjų. Čia yra išvardinti pagrindiniai bendradarbiavimo centrai, su kuriais teko dirbti per visą tą laiką, pagrindinėse tematikose, kaip puslaidininkiai, sensoriai, metalurgija, biomedžiagos ir taip toliau. Ir jūs matote, kad geografija labai plati, pradedant nuo Vilniaus, su daugybė mokslinių centrų Italijoje, Australija, Švedija ir taip toliau. Ir čia išvardinti tik pagrindiniai centrai, nes jų buvo ir daugiau. O dabar keletas eksperimentinių atvejų iš skirtingų tematikų, kurie iliustruoja, sakykime, mūsų laboratorijos svarbų indėlį į medžiagotyrą. Tai pirmas pavyzdys yra metalų superlydiniai, tai aišku yra metalurgija. Tie lydiniai yra labai brangus, naudojami daugiausiai aeronautikoje, gal dar kosmose ir panašiai, nes per brangus kasdieniniam pritaikymui. Čia jūs matote mikroskopiją vieno iš tų superlydinių. Jie visi praktiškai pagrįsti yra nikeliu, pagrindinis metalas yra nikelis. Ir matote, kad juose yra dvi fazės. Gamma ir gamma prime. Ir tos fazės išryškėja po cheminio ėsdinimo ir bet kurią mikroskopiją, ar elektroninę, ar atomic force mikroskopiją, matome struktūrą ir skirtingas fazes, kurių matmenys yra maži, yra apie 1-2 mikronai, ir reiškia mūsų turimi prietaisai tokios skiriamosios gebos nemato, nesugeba atlikti matavimų. Dėl to pagrindinius tyrimus atlikome sinchrodrone, kur skiriamoji geba yra 50 nanometrų ir galime matyti tokias struktūras. Čia vienas iš pavyzdžių, vieno superlydinio, kur matote daug įvairių metalų yra sudėta, jo mikroskopijos vaizdas ir cheminė mikroskopija atlikta sinchrotrone, kur matome tantalo cheminį vaizdą ir spektrus dviejuose skirtinguose taškuose, iš kurių puikiai matosi, kad cheminė sudėtis vienoje ir kitoje fazėje skiriasi. Tantalo ir wolframo santykis skiriasi. Dar įdomesnis tyrimas buvo šitų pačių superlydinių, įvairių superlydinių tyrimas po taip vadinamo creep test. Aš lietuviškai pats išverčiau, kad tai yra pastovios mechaninės apkrovos testas, bet nežinau, ar teisingai išverčiau. Reiškia, didelė mechaninė apkrova pastoviai ir praktiškai tas lydinys deformuojasi nuo apkrovos. Ir po apkrovos nebėra gražių stačiakampių, kaip buvo prieš šitą testą, pasidaro visai kitos geometrinės figūros ir čia matome du cheminius vaizdus, cheminės mikroskopijos rezultatus, superlydinį po šito testo. Vienas yra, jeigu aš gerai atsimenu, vienas yra renis, kitas yra tantalas. Toliau jau iš čia matosi, kad pasikeitė geometrija struktūros šitame lydinyje. O atliekant įvairių taškų spektroskopiją, matome, kad cheminė sudėtis skirtinguose taškuose, tamsiame, pilkame ar šviesiame, yra visiškai skirtinga. Reiškia, po mechaninio testo pasikeičia cheminė sudėtis šito superlydinio. Tai yra labai svarbus dalykas lydinių atsparumui ir taip toliau. Tiesiog, iki šitų tyrimų tai buvo nežinoma, tai reiktų pasakyti. Be to, kaip yra pabrėžta apačioj, visi kiti matavimai, mikroskopija ir panašiai atliekami po cheminio ėsdinimo, nes tik tada išryškėja struktūra. O fotoelektroninė mikroskopija leidžia be jokio cheminio ėsdinimo, kaip ir šiuo atveju, daryti cheminę mikroskopiją, matyti struktūrą, nes cheminė sudėtis skiriasi. Mums nereikia morfologijos kaip mikroskopijoje. Tai irgi yra labai didelis privalumas. Kita medžiaga, tai yra žemos darbo funkcijos sluoksniai, bario fluoridas, kuris buvo auginamas ant galio arsenido. Skirta tai yra, kaip jau anksčiau minėjau, termojoniniams ir fotoelektriniams celėms, kur yra kombinacija abiejų metodų. Tai reiškia, kad generuojama daug daugiau energijos ir europinis projektas, kuris prasidėjo 20 metais, baigėsi demonstraciniu modeliu, demonstraciniu prietaisu, kuris parodė praktiškai, kad prietaisas veikia ir labai gerai veikia. Ir tyrimai toliau tęsiami ir dabar naujuose projektuose, bet jie tęsiami tobulinimui šito prietaiso. Dabar, kaip jau sakiau, šituose prietaisuose labai svarbi yra darbo funkcija, kuri turi būti maža dėl elektronų generavimo ir dėl suderinimo su fotoelektrinė celė. Čia jūs matote juostinę diagramą. Kairiai pusėje yra taip vadinamas pozityvus elektronų giminingumas, kur vakumo lygis yra aukščiau už laidumo juostą, o dešiniai pusėje, kai kuriuose medžiagose yra sutinkamas retas reiškinys, kad laidumo juosta, kuri čia yra viršuje, yra virš vakumo lygio. Tai reiškia, elektronai jau savaime iš tos medžiagos yra emituojami. Ir aišku, kad tikslas yra turėti tokias medžiagas su neigiamu elektroniniu giminingumu. Ir vėlgi tai susiję, tos medžiagos visada turi labai žemą darbo funkciją, mažą darbo funkciją. Viena iš tų medžiagų yra bario fluoridas. Mes tyrėme daug įvairių medžiagų ir lantano heksaboridą, ir skandžio oksidą, ir dar bent 3 ar 4. Bet čia aš rodau patį geriausią rezultatą. Bario fluoridas reiškia yra termojoninės celės anodas, o galio arsenidas, kuris yra apačioj, jis jau yra fotoelektrinės celės dalis. Ir tyrėme, kaip keičiasi, auginant bario fluoridą, kaip keičiasi darbo funkcija, kaip jinai priklauso nuo sluoksnio storio. Pradedant nuo labai labai plonų sluoksnių, kurie net nesluoksniai, o salelės, ir toliau einant, didinant ir didinant storį. Rezultatas yra labai įdomus, nes čia matome darbo funkcijos minimumą, kažkur apie 2 nanometrus, tai reiškia būtų optimalus sluoksnis pagal darbo funkciją. Dar daugiau, atliekant valentinės zonos spektroskopiją, panaudojant ultravioletinę fotoemisijos spektroskopiją, matome, kad tai sutampa su neigiamu elektroniniu giminingumu. Šitas pikas, kurį matome iš pradžių mažą, o paskui didelį, jis atitinka 2 nanometrų storį, kur yra žema darbo funkcija. Reiškia, mes turime neigiamą elektroninį giminingumą ir tai yra ideali medžiaga, idealus sluoksnis šitų naujų celių kūrimui. Kita tematika, paskutinė, iš karto sakau, tai yra bidimensinės struktūros. Čia jūs matote visokius anglies alotropus, pagrindinius anglies alotropus, tai aišku ir deimantas, ir grafitas yra seniai žinomi alotropai, bet paskutiniais dešimtmečiais atsirado anglies nanovamzdeliai, grafenas, fulerenai, bidimensinės medžiagos. Ir yra, aišku, tarpinės medžiagos, kaip diamond-like carbon, kuriame yra 2 elektroninių konfiguracijų mišinys. Problema su šitom medžiagom, visom, tiek bidimensiniam, tiek ir ne, yra, kad paviršiaus analizės metodai neleidžia identifikuoti elektroninės konfiguracijos anglyje. Normalūs paviršiaus analizės metodai. Nes anglis turi vieną vienintelį karbon 1s piką ir abi šitos konfiguracijos sp2 ir sp3 jos sutampa tame pike. Reiškia, atskirti, kokią mes turime anglį, neįmanoma. Mūsų laboratorijoje mes pritaikėme nebe naują idėją, irgi italų laboratorijoje sugalvota prieš 30 metų, kaip matote, taip vadinamas D parametras, kurį skaičiuoja iš OŽ spektro anglies ir tai yra praktiškai OŽ piko plotis. Jis geriau matomas, kai padarom pirmą išvestinę, lengviau jį išmatuoti ir puikiai matome tai, ką jau buvo aptikę 91 metais, kad deimanto pikas yra labai siauras, parametras D yra mažas. Grafito jis yra daug didesnis, pikas yra platus. Reiškia, galima alotropus identifikuoti atliekant šitą spektroskopiją. Mes tą seną idėją plėtojame toliau, sukalibravome visą skalę nuo deimanto iki grafito įvairiems anglies bandiniams. Toliau pritaikėme ją grafenui. Ir čia aptikome labai įdomų reiškinį, kad jeigu grafenas yra matuojamas sužadinant su rentgeno spinduliais, su fotoelektroninė spektroskopija, tai jisai elgiasi kaip deimantas. Jo pikas yra siauras, parametras D yra mažas, kaip ir čia lentelėje parodyta. Jeigu mes sužadiname tą patį signalą su elektronų pluoštu, darome klasikinę OŽ spektroskopiją, staiga tas pikas pasidaro platus, kaip jūs matote žalią grafiką čia, ir taip pat matote lentelėje, kad jis pasidaro kaip grafite. Tai labai įdomus reiškinys ir teorinių požiūrių, ir jis paaiškinamas, yra padaryti ir teoriniai skaičiavimai, kodėl taip atsitinka, nes praktiškai valentinėje juostoje grafeno, kaip ir deimante, trūksta elektronų. O iš kitos pusės jis leidžia labai paprastai ir patikimai identifikuoti grafeną. Ir aišku, ne vien tik tai grafeną. Lygiai čia toliau yra tie patys spektrai, dar akivaizdžiau parodyti, kas atsitinka, kai sužadiname rentgeno spinduliais fotonais, kas atsitinka, kai sužadiname elektroniniu spinduliu. Reiškia, mes galim identifikuoti grafeną nesunkiai, bet lygiai taip pat galim identifikuoti ir anglies nanovamzdelius, ir fulerenus, nes jų praktiškai tik geometrinė forma kitokia, bet tai yra monosluoksnis anglies arba ten 2 sluoksniai ir taip toliau. Tai reiškia, turi labai didelį pritaikymą ir ypač sėkmingas tas taikymas yra kompozicinėse medžiagose, kur dedamas grafenas arba anglies nanovamzdeliai, nes šituo metodu nesunkiai galime pamatyti ir tą grafeną ir nanovamzdelius kompoziciniai medžiagui ir identifikuoti. Ir dabar ką? Pabaiga, apibendrinimas. Visa tai susiveda į virš 250 mokslinių publikacijų, 3 knygos tame tarpe, Hiršo faktorius, kuris žiūrint, kur pažiūrėsi, bet pakankamai didelis. Ir paskutinis leidinys, čia beveik reklamą darau savo paskutiniam leidiniui, tai yra knyga, tik ką išleista, World Scientific, kuriame yra mano indėlis, vienas knygos skyrius. Toliau belieka padėkoti savo kolegoms iš instituto Italijoje, daugybei kolegų Lietuvoje, su kuriais bendradarbiavome nuo pat mano pradžios Italijoje iki dabar, ir kolegos iš įvairių šalių, nuo Italijos iki Australijos. Taip pat labai dėkoju Jums už dėmesį.
Ačiū. Aš jau kolegai akademikui turiu daug klausimų, bet gal auditorija turi klausimų. Tai gal tada mes kuluaruose aptarsim. Ačiū labai. Dėkui.
Ačiū Jums.
Now we are reaching the high point, and due to the fact that in this room we have ambassadors, Your Excellencies, maybe you would like to say a few words. So in reverse order, we're starting from Italian side. So Ambassador Extraordinary and Plenipotentiary, His Excellency Mr. Diego Ungaro. Please.
Thank you very much for this fascinating opportunity, and to all of you for attending this event. I think that the scientific relations between Italy and Lithuania are personalized in the personality of Dr. Kačiulis and his work in Trieste. You may know that Trieste, since many decades, is concentrating a terrific level of knowledge and skills, and not only and strictly on the physics of materials, but including it. And I think that developing our cooperation among EU member states, we are strengthening our knowledge of the planet, of the physics of the planet, and developing the capacity to face increasing challenges and threats around the world. The political and geopolitical situation does not need to be commented tonight, but we are in a dire need to cooperate more in science to develop the capacity, the European capacity, vis-à-vis other major players, strategic players, which also are developing. I think that in the recent days we received information that the United Kingdom is joining again Horizon programs, and this is quite relevant because wisdom prevails on emotions and irrational, let me say, irrational components of politics, and Horizon is a major tool of developing what I'm saying, the European Union capacities in these fields. So tonight we are celebrating through this fascinating presentation of Dr. Kačiulis and of the whole event, these areas of activities. I wish to the Academy of Sciences of Lithuania best results going in this direction, as the Italian counterpart of the CNR, the Council of National Researches, which is very much scattered around Italy, and Dr. Kačiulis presented the different sections and different bodies that are involved in our entity. So it's all over Italy, not only Trieste, but many other towns. So there is no physical place for our institution. But let's go ahead together and let's develop the European skills. Well, of course, even science, like culture in general, is very international, and he mentioned that there are cooperation even beyond European borders. Don't take me too strictly on the European Union borders, but I'm sure that I share the message that we have to strengthen the European Union capacity with the cooperation, and the UK is an example, even non-European Union members, in order just to stay in the competition with the proper skills. Thank you to everybody.
Thank you very much. Grazie. Grazie, it's in Italian. Okay. So now I would like to invite His Excellency Ambassador Extraordinary and Plenipotentiary of the Republic of Kazakhstan, Timur Urazayev. Please.
Ladies and gentlemen, firstly, I'd like to thank President Banis for inviting our delegation to, and I congratulate you with such a significant event like the signing of bilateral memorandum for further cooperation between the National Academies of Science of Kazakhstan and Lithuania. I think that our historical and traditional ties between the scientific community of Kazakhstan and Lithuania will further develop in sake of the science and stability. As one of our colleagues said that we all dream to make the world better, and he enumerated 4 features: health, stable, if I'm not mistaken, health, stable, wiser, and someone. I'm sorry, I don't remember. So science is fulfilling its duty for health and stable. As for diplomats and politicians, secure, the force is secure. Security and secure. But politicians and diplomats, I think, has not yet fulfilled its own task to make it secure world. And I'm very glad that all the scientists, not only in Lithuania, Kazakhstan, Italy, and all over the world, they serve for the very sacred purpose to make this world better. I congratulate Madame Zakaria with this event and starting the practical cooperation between 2 countries. Apropos, Madame Zakaria is traveling further to Latvia. That's why I apologize for fluff. And I congratulate the General Assembly of the Academy of Science of Lithuania with this fascinating event. I hope that science will serve for all of us for a better world, and hope to see you next time, next year, with new achievements and new scientific openness. Thank you.
Thank you very much. And before coffee, which is waiting outside, I believe, it's the last duty. I invite Your Excellencies ambassadors, new foreign members, President of Kazakh Academy of Sciences and Vice President of Kazakh Academy of Sciences to take a joint picture here for the history.